Good morning, everybody. Thank you for coming to our session. We are here to celebrate and critique one of the major developments in science of recent years, which is gene editing. And in the course of that, I hope we'll have some discussion on a number of issues connected to that topic and to scientific developments that pertain to that at large. We have a most august panel here to join and help us in this conversation. Quick introduction for me. My name is John Thomas. I'm the chairman of the California Institute for Regenerative Medicine, which for those of you who are from California was the agency set up by Proposition 71 in 2004 to fund stem cell research with $3 billion of state money throughout the state of California. Introducing my fellow panelists, starting on my left, your right, we have Hank Greeley, the director for the Center for Law and the Biosciences at Stanford Law School. Immediately to his right, we have Dr. Amanda Clark, professor and vice chair in the Department of Molecular Cell and Developmental Biology at UCLA. To my right, we have Dr. David Baltimore, Nobel laureate, president emeritus, and Robert Andrews Millikan professor of biology at Caltech. And to his right, Dr. David Agus, professor of medicine and engineering at USC and the director of USC Center for Applied Molecular Medicine, and an author, has had a number of best-selling books, most recently one called The Lucky Years, which I hope he will comment on in the course of answering some of the questions that we will be discussing. So getting straight into the substance here, I thought to paint the context of the times, this has been called one of the golden eras in biological and medical research, and David's book, The Lucky Years, touches on that. So I thought I'd ask him first to give a little comment on sort of the overview of what's happening in medical research and why this is such an exciting time that we are all living in and experiencing. David.
Thank you. It's a privilege to be here. So 13 months ago, President Jimmy Carter announces that he has melanoma, skin cancer that went to the brain. And that literally was a death sentence several years prior. And he received a therapy that blocked the don't eat me signal on his cancer. And his own immune system put his cancer in check, so much so that 3 weeks ago he announced he's cancer-free and no longer on treatment. So this is a new era of medicine, an era where we can harness the immune system to attack disease, an era where every patient I see with cancer, we sequence their cancer prior to me seeing them. And I know many times the on and the off or the signaling pathways within the cell. And so there literally is a new discovery happening every week, every month. And that's very different than the pace of science in the 90s and the 80s, et cetera. That pace has changed and quickened, and I think you'll sense excitement among all our panel around it. But to set the stage, I do want to tell you a story. And this is a story that started in the 1990s when the government gave a grant to identify the mutant gene for breast cancer called BRCA. And we all saw several years ago, Angelina Jolie, proudly said, and was a hero for being transparent, I have this faulty gene that allowed me to have a higher predisposition for breast cancer and ovarian cancer. So the New York Times asked that we write a piece the next day. And what I wrote about was that when this came out, it was licensed to a company that charged $5,000 to $6,000 for this test. And so for a period of 15, 16 years, you had to pay a ransom to see your own DNA. You paid $5,000, $6,000, they would tell you the answer, whether you had this or not. Well, tens of thousands of people a year couldn't afford that. And many of them died from having the disease they couldn't identify early. Well, about a year and a half ago, this US Supreme Court said, or 2 years ago said, you can no longer patent a gene in our country. They had, in a sense, democratized DNA. And there was a lot of clapping on the back and cheering. We've done it. Now that test is less than $200 from $6,000. The problem is that in that interval, 16 years, tens of thousands of women died. And so with technology happening at a pace that's unprecedented, we need the legal system, the ethical system, the moral system, our ways of thinking to catch up, because for every breakthrough, we don't have 15 years to change how we think.
Thank you. And by the way, David's being modest. He is one of President Carter's oncologists and is integral to the treatment that he just described, which is nothing short of remarkable. In the context of the many developments going on in science, gene editing is one of the hottest. I don't know how many of you were here for the Monday lunch session and heard Jennifer Doudna talk about CRISPR-Cas9 and how she came up with that and the application. But I thought what we would do is to have Dr. Baltimore talk us through exactly what gene editing is in plain English, so that you'll be able to better follow exactly what the discussion is going to be about. So, David.
So gene editing is about changing the sequence of your genes, of anybody's genes, of any animal, plant, bacterium's genes. And we want to do this because sometimes we don't have an optimal sequence in our DNA. So I'm going to assume that we all know that human heredity is carried by DNA, which is a long polymeric molecule, that it's found in the nucleus of all of our cells, and that it's organized into chromosomes, which are in turn organized into genes. And so when we talk about gene editing, we talk about changing the code written in DNA. And if I could have slide 5, which sort of says that. And so if we want to change the sequence in DNA, what we have to do is find the sequence we want. And there are 3 billion base pairs of DNA in the nucleus of each of our cells. So we have to search among all of that for this one sequence where there's one change that we want to make. And then we want to cut at that point and alter the sequence. But cutting is key. So we need to be able to identify the spot on the DNA, and we need to be able to cut the DNA. Cutting DNA is pretty straightforward. We have lots and lots of what we call nucleases, proteins in our cells that cut DNA. So we can put in one of those and cut the DNA. Problem is, how do you identify the place you want to be? And so we started solving that problem now a number of years ago with something first called zinc fingers and something later called talons, which are proteins that can recognize the sequence in DNA. But proteins recognizing sequence in DNA is clumsy. And so these things were a little clunky. They have their use, particularly talons have pretty wide use in gene editing, but they were not the revolution that we now have with CRISPR-Cas9 and that Jennifer Doudna played such an important role in developing. So if I could have the next slide. So protein recognition is all well and good, but we have a fabulous way of recognizing sequence in DNA. And that's another DNA molecule or an RNA molecule, which is fundamentally the same thing. And if you know the sequence that you're looking for, you can make the complement of that sequence trivially in the lab and then put it in, and it will be found by a DNA or RNA molecule. It's the Watson-Crick complementarity principle at work, discovered years ago by Watson and Crick, and for which they are highly honored. So actually what we want is a combination. We want to be able to recognize the sequence with DNA or RNA. It's RNA that we'll use. And we want to be able to cut with a protein. So we need, if I could have the next slide, to be able to do that. And what we do is we make a combination of RNA and protein. CRISPR is RNA, Cas9 is protein. And in that slide, the protein is the sort of eminence grise in the background, and the RNA is the string of pretty colors. And what it has done is found a sequence in the DNA molecule that runs along the bottom there. And that's the whole thing. It finds the sequence, and now it tells the nuclease that it's attached to cut at that place. Next slide. So why do we want to cut? Well, if you cut both strands of DNA in the nucleus of your cells, it's a disastrous thing. There are all sorts of systems in the cell looking for that to happen and ready to repair it because it can kill that cell. And so it repairs it. And in the process of repairing it, it can either make a mistake and inactivate the gene that you're in by cutting out a few extra base pairs, or it can be guided by a guide DNA to make an exactly appropriate change. So let's say we're looking at a cell that has a gene for sickle cell disease. It can find that gene and change the one nucleotide that makes it from a normal gene into a disease-causing gene and put it back to normal. So that's the principle of what we're working with. The next slide. The CRISPR-Cas9 then is a tool to edit a genome. It could be a human genome, it could be an animal genome, it could be a plant genome. If it's a human genome, that it will make a permanent change in the heredity of whatever person might be born from that changed gene. Particularly if you're if you're dealing with the germline, the eggs and the sperm that give rise to the next generation. How often would you want to do that? Just one last slide. The answer is fairly frequently. There are 6,000 monogenic traits in diseases that we know about that we'd want to change, and they run through all of the various parts of the body. So to quote Eric Lander, if I could have the last slide, genome editing is a method to correct typographical errors. They're the kind of error that we inherit, and we want to get rid of them so that our offspring in particular can be normal.
Thank you, David. I wanted to highlight, if you could just elaborate for 1 second.
Yes.
You used the term monogenic.
Yes.
And why is that a critical word as opposed to diseases that have multi-genes?
Well, actually, the next slide. 12, yes. So monogenic diseases, which are represented on the right there, are rare traits that where a single gene with a single defect in it of some sort can cause a disease, often a lethal disease. But there are many aspects to human life, like height and obesity and heart disease, which are caused by many, many different genes, sometimes genes acting in concert with each other. And for those, this technology is relevant, but it's going to be more complicated. So that's another step in the development of the technology that you can think about. I focused in my discussion only on the simplest example. And there may be things that we wouldn't call diseases. Well, height is not a disease. That we want to change. People want taller children that come from short families like mine. And to do that requires changing lots of genes. It's more complicated and it's more ethically charged because it's society based. It's not a disease. It's a choice of people to have taller children or prettier children or smarter children, all of those kinds of things.
Okay, thank you. By the way, anybody in the audience, can you identify the two gentlemen on the picture on the right?
Shoemaker and is that Wilt?
Excellent, very good.
Wilt
Willie
Chamberlain.
Shoemaker and Wilt Chamberlain.
Right.
Okay, so thank you very much, David, for that description of the editing process. He touched on sort of what is really the really hot issue, which is the so-called germline gene editing, which is the thing that carries many of the ethical issues that we're going to discuss. I'm going to ask Dr. Clark if she just do a brief elaboration on germline gene editing to further set the table.
So can I have slide number 1, please? So it looks like, as I look around the room, that perhaps many of you haven't taken a high school biology class, perhaps for a number of years. And so I just want to introduce you to what the germline is, and just to remind you that there are trillions of cells within our bodies, and a very, very small fraction of them have the ability to pass our DNA onto our children. And this is where the ethics of gene editing is really at its pinnacle. And so the germline cells are the cells in our body that are made in the testis if you're a man, or the ovary if you're a woman. And these are the cells, and they are the only cells able to do this, which can pass our DNA on from us to our children. And so what you're looking at here is a beautiful pseudo-colored image. This is not the colors that they are in the body. The large yellow structure is an egg cell, and the little blue, I guess, ribbon that you can see, that is a sperm cell that is just about to fertilize that egg. And so can I have the next slide after this, number 2? So at the point of fertilization, if I was working in an in vitro fertilization lab and I was looking down the microscope, these are the kinds of images that I would see. So the fertilization of an egg and a sperm cell leads to something called a one-cell embryo. And this embryo now has the genome, or the chromosomes, that came from mum through her egg and dad through his sperm. And so in the top corner, you can see that big round circle and perhaps 2 smaller circles on the inside. They are the genomes of mum's genome and dad's genome that are starting to merge together in the middle. And then that egg cell with its complete genome will start to divide to form a small embryo. And so you can see lots of bubbles. It looks like a ball of grapes. So those are the cells that are ultimately going to make the placental cells, as well as the cells that will form the embryo at implantation. So at the very bottom on the right is a human embryo. It's a blastocyst embryo that is hatching from a protein shell which protects it in the lab, in the IVF clinic, as well as in the female body. And that embryo will hatch and ultimately implant into a womb. Can I have the next slide? And so when we're talking about germline genome editing, what we're talking about is not the trillions of somatic cells in our body that make up our liver or our lungs, our brain, other cells you can think about. We're just talking about the cells of the germline, the egg and sperm cells, and then also that window of development which can be accessed in the in vitro fertilization lab, which is the growth of human embryos. And so on this slide here, what I'm illustrating is that, can you not go to the next slide, but push a button to highlight something, perhaps? Is that possible? I put an animation on here, so no? Yes, great, well done. So what I'm showing here is what we're talking about with germline genome editing is using the tools that Dr. Baltimore talked about, these very precise genome editing tools to change the DNA of the egg cells or of the cells of the early pre-implantation embryo. So this would be occurring outside of the body in a laboratory. And if this was an edit that was going to be passed onto our children and then their children's children, the embryo would be implanted into a womb, and then that would give rise to a baby which has that offspring would have all the traits of the parents plus that precisely edited gene, which the goal would be to edit a gene that causes a disease so that it no longer causes a disease and that you can have a healthy baby. Can I have the next slide? There is another approach that is being used today, not in the United States so far. This work has been published in other countries around the world and has been approved in the United Kingdom through their specific regulatory body, which regulates the use of eggs and sperm and embryos in IVF clinics, as well as for research purposes. And this is to use germline genome editing to edit the egg and the sperm cells and the human embryos, and not to transfer those embryos into a woman for the purpose of having a baby, but instead to use these edited embryos to understand whether genome editing will even work. We don't know this yet, whether genome editing will really work in the egg and sperm and early embryo, as well as to understand the basic biological principles and how an embryo forms, how the placenta forms, in order to help us improve existing therapies such as in vitro fertilization or child health during prenatal life. And those sorts of experiments are done without transmitting that gene-edited change onto the next generation. So these are the two different ways of thinking about germline genome editing.
Thank you, Amanda. Of germline genome editing, Hank, you were quoted as saying, what was impractical is now almost every day. That's a big deal. Why is that? And can you lead us into a discussion of the ethical issues surrounding germline gene editing?
Sure, happy to. And I think part of it is historical. If we go back 40 years, or actually 41 years, to the first big public controversy around DNA editing, it involved a meeting at Asilomar State Park and Conference Center in Northern California, co-chaired or co-organized by David Baltimore here, that talked about recombinant DNA and how we should go forward with recombinant DNA. There's a lot of concern in the public at that time about messing at all with DNA. And many people said at the time, well, don't worry, we will never change the human germline genome. We'll never make inherited changes in people. We'll only change things in living people. That was an easy promise to make 40 years ago because nobody had any plausible way of actually doing embryo gene editing or gamete gene editing at that point. What's happened subsequently is now it's plausible. And the question is coming up again. There is a large set of people in the bioethics world who think that germline editing should be a red line that we should never cross. Because if you were to change my genes to make the rest of my hair turn white, get this little mustache and a little bit in the eyebrow to turn the good color, that dies with me. If you change my sperm and I have a baby, that goes on to the next generation and potentially to the next generation forever, or at least as long as our species survives. And so people say we shouldn't make that step. I actually think the case for that isn't as strong as it is often asserted to be. How many of you are parents? Okay. How many of you had your children turn out exactly the way you expected? Yeah, that's the right number. The genes you pass, the DNA you pass down to your children is not exactly the same DNA you received from your parents. Mutations happen during your lifetime and during the development of the child. The human germline is constantly changing, but it's changing in a random and sometimes negative way. I think we should be worried not so much about whether we're changing the next generation's genes, but A, whether it's safe, a really important ethical issue, and B, what purpose are we changing them for? If a couple, let's say two people with cystic fibrosis, fall in love, get married, want to have children. Right now, any children they had would themselves have cystic fibrosis. You could use germline gene editing to give them the chance to have children who did not have cystic fibrosis. And I think the ethical case against that is pretty hard to make. But that's where the current battle line is. There's a lot of draw a red line at germline, draw a red line at anything crossing generations, and things that only affect living people, worry less about.
Thanks, Hank. You referenced, and by the way, I hope the rest of the panel would like to weigh in on the ethical issues But before we get to that, you referenced Asilomar and David's role in that. There have been similar meetings of scientific bodies around the world in the advent of gene editing for germline cells that have addressed the question of a moratorium on this particular technique. And David is sort of singularly positioned to be able to discuss Asilomar and recombinant DNA, what the issues were, and the meetings now that they've had around the world in connection with gene editing. And I thought it would be informative for everybody here to get a little perspective on what was different and what isn't different, and David's role in all of this. So David, if you could elaborate on that, please.
Well, the Asilomar meeting was largely a meeting of scientists. We also invited and had attendance from lawyers, the press, and others, but the discussion was mostly among scientists. And it was about safety. As Hank said, safety becomes an ethical issue when it can affect, when unsafe behavior can affect other people. And that's what we worried about. And we said we didn't know enough to be sure of the safety. And so we said there's certain things that we shouldn't do at this point, and there were other things that we should develop slowly with careful concern over whether the safety was there. And that's exactly what happened. So over the next few years, the technology was applied to more and more complicated situations with new safety issues associated with them. We ultimately proved that it was pretty safe and that it could be used very widely and is used very widely today and has been the core of the revolution that we've seen in modern biology and in the core of the biotechnology industry. When CRISPR-Cas9 came along, it sounded like a parallel situation, and it is a parallel situation. New technology, new questions have to be dealt with carefully. We asked again that people not run forward with this technology, but rather think carefully through it. And this time called for an international body. Previously, it was national bodies that handled it. And that international body is in the process of formation under the auspices of the American, U.S. Academy of Sciences, U.S. Academy of Medicine, and the Royal Society and the Chinese Academy of Sciences, which came together to have a meeting last December, the end result of that meeting being a statement that we should not move ahead without careful consideration of the safety and now the ethical problems. Because whereas at Asilomar, as Hank said, we could sweep those under the rug, we no longer can, because we are dealing with human heredity. And I think we need a wide consensus and strong international norms to decide that there is a line that we are willing to cross. I would hope that that line is the kind of line that Hank described, giving the people the opportunity to have children that are free of disease. Should we go further and worry about enhancement, as I described it? I think that is a major challenge to our thinking. And I, for one, hope that we have a deep discussion about that. But it is very hard to imagine that there will be an international agreement that there's a bar we should never cross. Because there's more and more opportunity. It's going to get easier and easier, safer and safer. And there is going to come a time when we have to think carefully about actually crafting the next generation of humans rather than allowing the genetic lottery to give us whatever we get.
So David Agus, what, in your opinion, Hank and David Baltimore have touched on sort of bad uses, etc. What in your opinion are good uses and bad uses of this technology?
The potential for this technology is staggering, as was alluded to. You can change T cells so that HIV or the AIDS virus can't get inside them. You could potentially re-engineer those T cells to do what you want in the body with this technology. Many of the diseases that the other David referred to, we can, yes, have an impact on. But I think it's been made, the technology is not perfect yet. It's not totally controllable yet. And I think the important point that I want to put out there is it's a different world than Asilomar. The world is flat at the present time. And so you may get China, the UK, and the US together, and the scientists say, hey, listen, we're going to do X, Y, and Z. But what's to stop a scientist from moving to another country, some lesser developed country, and starting to do this? There is no body that has the ability to enforce things across the world. And in today's world, to set up a lab to do this is not that difficult. So I do think we need to, at the same time we talk about the legal and the ethics, we need to educate. Because in order to get normative behavior change, you need leadership. And with leadership, you need that education. And so I do think we need these discussions to be not just in private, but in public. And we need to start to understand the ramifications. The bottom line is we are a complex emergent system. When you change one thing, other things change also. And in order to do the experiment right, you need to look 50 years down the road or 70 years down the road and say, what happened? Well, obviously, we're not going to be able to do that. So there's inherent risks to all of this. But I think what gets all of us excited are the potential. I live on a daily basis and see suffering with disease. Patients come to see me every day, and they say, please accelerate things. I need it. And so, me on those front lines, I'll do whatever I can to push the technology forward. But I love what David and others are doing of getting that discourse going, because it really needs to accelerate, because this technology will get better and better, and the applications are going to grow.
A question for the scientists on the panel. Are there any issues with CRISPR-Cas9 that give you pause, such as off-target editing or, I use the term inefficient yield, which means you try something a number of times and it only works a limited percentage of that? Anybody want to comment on that?
The answer is yes.
Yes. The answer is yes. We're worried.
Okay.
Right.
No question about it. The holy grail for this, right? I love germline editing, and I think there's a potential there. But what if you could take this remarkable package and deliver it to a cell in the body or certain cells in the body and change things post-germline when we're grown up or when a child has an issue and fix it? So the ability of not just to fix that early embryo cell, but to actually address issues as they happen is something that over time we're going to get better at also.
Amanda, do you want to comment on off-targeting issues?
Right, sure. So I don't want to put words in the mouth of the other scientists on this panel, but I think that the genome editing of the cells in the adult body is, of course, much less controversial than the cells of the germline. And there already is gene therapies today for somatic cells in our body, as David pointed out, in order to provide a therapy for HIV or to change the genes in our blood cells in order to prevent terrible diseases such as the bubble boy syndrome. So these are all targetable therapies in our somatic cells. In the germline, what we know so far is from 2 studies that have been published, and both of these studies were performed in China. And this was using human embryos that were donated to research. So they were created for the purpose of reproduction, and when the family had finished using the embryos for their family planning purposes, they made that difficult choice of donating the embryos to research versus donating them to another couple, or having the embryos discarded, which is another choice, or leaving them in storage. And so, so what we do know is that when CRISPR has been attempted in human embryos, this is for research purposes, this is not to transfer the embryo back into a woman to establish a pregnancy, is that CRISPR has off-target effects. And what that means, it changes the genome in unanticipated places. So does that mean that we should stop germline genome editing today? It doesn't work, it's all over. So I would argue no. I would argue that this has taught us something very important about the state of the technology today, and for us to think about ways to change the technology to make it more precise so we can harness it to do what we want it to do. But right now in the experiments in human embryos, this has proved to have off-target effects, and therefore very clearly illustrates this technology is not ready for use in therapies for overcoming disease.
Let me add one point in defense of the Chinese scientists who did these two papers. They used human embryos that could not become babies, that had been fertilized by 2 sperms, so they had 3 sets of DNA instead of the normal 2 sets, and could not be viable. And for people who are concerned about the possibility of some of these edited embryos becoming babies, that's an important line.
But it also might have been the result of the failure, because
Yes.
normal embryos don't have 3 nuclei. So how do we know that we don't have an overactive repair machinery that's causing the problem?
Which is the great dilemma. The closer you get to working with real humans, the more the ethical issues are, but the farther away you are from getting with humans, dealing with real humans or viable humans, the less certain you can be of how applicable your results will be.
There are more than 1 million embryos in storage within the United States from couples that are accessing IVF to overcome infertility. And so what's going to happen to those 1 million embryos or more? Because IVF and treatments for infertility is a very highly accessed therapy in this country. It's been estimated that more than 1% of children in the US born today are born as a consequence of IVF. And so couples in the United States do donate their embryos for research purposes because they feel that that gift to scientists could help somebody else not go through the problems with infertility that they have had, or can help develop a cure through stem cell therapy. And so there are a lot of normal embryos that will be donated to research, and these could be used in order for scientists to figure out is this therapy even going to work or not on a healthy embryo. And in a study that was performed at Stanford, if you ask couples that donate their embryos to research purposes, what do you want your embryos used for? Do you want them to be used for stem cell research or to be used to understand human embryo development? About 75% of donors of these embryos will say, we don't mind. You can use it for stem cell research or to understand human embryo development. So at least for the couples that are going to donate these very precious resources, there is an understanding that these embryos will be used to help further the knowledge of human embryo development or potentially new technologies such as CRISPR, so we can make an educated decision of whether this technology is going to work or not. We can't make that decision right now.
I will make the point, which I think is important, though, is that when we talk about these esoteric diseases that very few in the room can identify with boy in the bubble syndrome or a particular sickle cell anemia. But all of that research that we're doing there is also applied to every other disease, not to the germline. So we can make a mouse model that used to take years. We can do that in weeks now. We can turn off a pathway in a cancer cell and see how it affects the cancer, which can change treatment for a particular cancer literally over several months. And so very exciting, the applications and the cross applications from the germline work to other areas of work. So in a sense, the benefit is not just to these esoteric diseases, but it really is to the population at large.
So Amanda talked about the China work done. That brings up the general question or point, which is this is going to happen someplace. And the question is, why shouldn't the U.S. be out in front in work to develop and refine the uses? David, I ask you that question. David Baltimore.
Well, I think the U.S. should be out in front. Actually, modern biology is a creation of U.S. science, and we have the tradition of being the place where innovation occurs. In this area, we are stymied, and we're stymied by congressional action, so that what Amanda said, which is the donation of embryos for experimental purposes, is illegal in this country. Now, the legality of it is something I can never keep totally straight. And so I'm going to ask Hank if he could put that in a couple of sentences.
Sure. It's not so much that it's illegal to use federal funds to do research with embryos that may destroy or damage them. Some states make it illegal. In Louisiana, you go to jail if you destroy an embryo. So with Hurricane Katrina, there were boats sent to pick up vats full of frozen embryos to avoid liability for the IVF clinics. But for the most part, it's not that it's illegal exactly, it's that you can't use federal funding for it. But if you can't use federal funding for it, that's a major constraint.
All right, I will note, those of you note the history of my particular agency, that it was President Bush's ban on federal funding for generating new embryonic stem cell lines that was the impetus for Prop 71 that formed the California Institute for Regenerative Medicine. So there are ways around it, although that's a pretty dramatic move, yes.
And let me add one more footnote to my answer to David Baltimore's question. In the last Budget Reconciliation Act passed in December, Congress also, in its wisdom, decided that the FDA could not approve any clinical trials with edited embryos. They didn't define the terms very clearly, but if we ever got to the point of clinical trials, which we won't in the 1-year span of that budget bill, that would also be a barrier, and a significant barrier in the United States. And if that rider keeps going year after year, as the Dickey-Wicker Amendment with respect to embryo research has for the last 20-some years, that will be a substantial constraint eventually.
Actually, I think they said that the FDA may not consider
Yeah.
an application.
Yeah.
They can't open the letter.
It was actually quite funny because the original language said the FDA couldn't spend any money on it. And I noticed, and other people noticed, if you want to do research with humans, you file something called a request for an investigational new drug exemption with the FDA. If the FDA does nothing, you're allowed within 30 days to start your experiment. So when they said the FDA couldn't spend any money on it, what that meant was the FDA couldn't block it. They then amended the law to say any letter, any request for such permission to the FDA shall be deemed not received. And as a lawyer, what can I say? I'm sorry.
But you know, the field is taking off. The American Society of Human Genetics is on the East Coast this week. 13% of the abstracts at the American Society of Human Genetics involve the use of CRISPR.
And increasing. Yeah. Amanda, you referenced IVF work. So one of the suggestions, given the ethical concerns about CRISPR-Cas9, is why employ that technology? Why not achieve the same result through IVF and genetic screening to get the desired result?
Well, of course, the altering the genome of an egg or an embryo would not be the first approach that would be taken to be able to enable a couple to have a healthy child. So there are existing technology that's used, such as pre-implantation genetic screening for couples that are known to carry a disease gene and they want to have a healthy child. And so in IVF clinics today, they will screen embryos to only pick those embryos to use for reproductive purposes that will result in a healthy child, and select away the embryos that carry the disease genes. But a lot of individuals who access IVF don't make very many gametes. So sometimes you don't have very many embryos to choose from. You might have only 2 embryos to choose from. If you're my age, you might only have 1 embryo to choose from, just because as we get older, we don't make high quality gametes anymore. The risk of them carrying additional diseases, disease genes, becomes higher and higher. And so of course, changing the genome itself is not something that you would do first, without first selecting for embryos to see if there are alternative ways that a couple can have a healthy child. So this would be something that would help a relatively small number of people, but I think as a discussion in a society, just because the number of people that would be potentially benefit from a genome editing technology is relatively small, does that mean we should not even try and attempt to see if this technology will be useful? Should we exclude them from a potential therapy today because we don't know whether it would work or not? So that's my opinion.
So we've referenced the impact of Congress in the field of the FDA. We could spend a lot of time discussing, once this becomes a more accepted practice, what's the process of getting the FDA to speed up its internal process to get to where you'd be in a position to implement this technique quicker, but leave that for another discussion. So we talked about Congress, FDA, no Milken Institute panel would be complete without somehow asking at least one question on the presidential election. So for the panel, I would like to ask, how do you think the technology of gene editing and other advanced scientific techniques will fare under President Clinton or President Trump?
I mean, can we edit President Trump into a...
An acceptable form.
You can edit his hair.
He already has.
Okay, anybody want to take a, take
I-
a stab at that?
I believe that Secretary Clinton has a very, I know she has a very strong interest in biomedical research, in the application of biomedical research, and I am confident that she will make every effort that she can. After all, Congress will still be there to see that this moves forward. What happens on the Republican side, I have no idea.
Anybody want to take a stab at that? Hank, you like sticking your neck out.
I don't have a clue what a President Trump would do with this. It's not clear to me that a President Trump has a clue what he would do with it, or it would be the same clue today as it would be tomorrow. So I think I haven't seen any real policy statements from him addressing this set of issues, and his overall approach to the Republican base is so quirky that I really don't have a clue what he would end up doing as president. Except that, as David Baltimore pointed out, any president is going to have to deal with a Congress for which these issues can be quite important.
We've all heard the term precision medicine thrown around as sort of the next big thing in medical treatment. How do we think that gene editing will factor into the precision medicine revolution? David, you want to try that one?
Precision medicine is, the simple concept of the right drug for the right patient at the right time. And we can say that in those terms, but actually getting it to fine grain and what that means is hard. So cancer patients now, we can routinely look at the signaling and say, where did the DNA go awry to allow those cells to continue to live? Can we use gene editing there? Probably not. Particular viruses, we know exactly how they get into a cell, so maybe we can change cells so those viruses can't get in, the ones that are potentially not amenable to vaccine, and that's certainly a way we can approach this issue here. But the advances in our space and the ability to target DNA is going to be interesting because we now starting to know what are predisposition genes. So I can look at your DNA and say you've got a higher than average likelihood of getting Alzheimer's disease or of getting disease X or disease Y. And so the challenge will be going forward, as David alluded to, diseases that have multiple genes contributing to it. How does this figure in? And it obviously first happened in the research world. The application clinically is probably decades away. But it's certainly something that it's exciting to think about. But the application and the models that we all study, who knows what it's going to yield today?
David?
Precision. There is nothing that gives you more precision than the complementarity between two DNA or RNA molecules. And so what really, when we say we want to minimize off-target effects, what we're talking about is precision. And we know how to measure it, and we know how to be certain of it. And so the kind of technology we're talking about is the ultimate in precision medicine. And will be used somatically, as David has extensively discussed, and I am absolutely certain it will be, with enormous effect, partly because we can target so precisely.
Mander, do you have a thought to add on that?
So I think attaining the precision of what we all are trying to achieve in precision medicine comes from very strong basic research. So as I'm a basic scientist, I'm not a clinician. And in a basic science research lab, we try every day to perform the best experiment possible that can be reproduced day after day after day. And so getting to the precision of precision medicine with genome editing requires very strong support of basic science. And this comes back to the previous question in terms of the next president of the United States. We need somebody who has a thorough understanding of the importance of basic science research in enabling these precision therapies to move to patients.
Question for you, Hank. So years ago when IVF came around, it was hugely controversial, and now it's every day. When Dolly the sheep was cloned in the mid-90s, the topic of human cloning was massively controversial, and yet a couple years ago you actually had successful human cloning at the Oregon Health and Science University up in Portland. How many here in the room knew that that happened? Anybody raise their hand? Okay, that was about 3 people in the audience on a topic that was wildly controversial 20 years ago. How far out into the future will it be that germline gene editing will be thought of as commonplace, and it will be something else that'll be the next controversial thing?
Well, so to prove that I actually am trained as a lawyer, I'll give the first 2 words that any lawyer should give answering any question. It depends. It depends in part on how safe and successful any sort of human germline editing turns out to be. It also depends on whether in the next 5 to 10 years it becomes possible for people to do the basic research that might lead to safe and effective human germline editing. I do think that if it were used just to fix diseases, to prevent diseases in people for whom there was no other good way for them to have children without the disease, it would ultimately be relatively uncontroversial and something else would take its place. If, on the other hand, it's used to create never before seen in human hair color in presidential candidates, or other forms of enhancement or disenhancement, then I think that controversy is probably going to be with us for quite a while. The enhancement controversy, I think, will not go away soon.
Okay, I ordinarily would ask the rest of the panelists to give their thoughts on that, but we're down to about 5 minutes, and I wanted to hit on something else for the audience, which is other uses of CRISPR. We've talked about it with germline gene editing. There are many other things that it can possibly be used for, because basically it works on any living organism. I will note, for example, that there is considerable work being done at various places in the country on what they call driving maleness in mosquitoes that are the mosquitoes that are transmitting Zika virus, malaria, dengue fever, et cetera, and they're thinking about a vast population of these mosquitoes that are gene edited to go out and mate with mosquitoes in the wild and basically preclude them from producing offspring. And that is because Zika is such a big topic, and the last Milken Institute meeting was on global public health in Washington, D.C., and Tom Frieden from the CDC spent a whole lunch session on Zika virus, which was very interesting and, quite frankly, very disconcerting. But they are looking at alteration of the mosquitoes as one alternative use. David Agus, in your book, The Lucky Years, you commented on research being done on the killifish and aging. Could you just talk about that for a minute here for the audience?
For the record, I'm against Zika virus. You know, but mosquitoes have killed more people on this continent, in this world, than all wars combined. And so it's an issue we have to address, and we have to use science in a powerful way to address here. Because what's going to happen this summer, as there are, puddles of water across the country and little mosquitoes are happy there, is any woman who's pregnant or thinking of getting pregnant who gets bitten by a mosquito is going to have that fear, and it's a realistic fear that that child will be born without a formed brain. And so this is something that is going to come to the forefront of every major news media, paper, et cetera, as the temperature warms across the country. The notion of using these technologies to make small organisms like this fish refer to, live longer is very powerful. This technology can be used to make corn so it doesn't get pests. This technology can be used to tomatoes so they don't rot on the way to market. And right now, 30% of the produce going in our country is lost during transport. So there's a potential here to use science for good, and at the same time, think of the health implications of what they're doing. In order to do that, and I know we're nearing the end, is I would just push to develop data standards for ways of describing what we're doing so that scientists can talk to each other, can talk to regulators, et cetera, and to push for building the framework to allow this to happen, both on agriculture, on food, and on humans. Because right now it doesn't exist. There's a lot of debate over the ethics, but very little over those standards which allow us to study and learn from every experience.
David?
David said it.
Okay, thank you. Anybody, any of the other panelists want to speak there?
Yeah, this is actually what I lose sleep over. The human side of it, you'll need an IVF lab, you'll need a doctor, you'll need a willing mother, you'll need FDA approval, you'll need IRB, ethics committee approval, there will be tort lawyers circling around. The human stuff is relatively controlled. What makes CRISPR so exciting is it's so cheap and easy. Two people with BS degrees in biology and a garage and $1,000 and access to mosquito larvae could, in theory, change all of the mosquitoes of a particular species in California in the space of a year. And we don't have a regulatory system that exists well enough to discriminate between good uses, like perhaps stopping Zika virus, and uses that may seem good but turn out to be terrible, let alone a system that might be able to stop a terrorist from turning mousepox into smallpox, or the benign and ubiquitous form of E. coli, which is in all of us, into the pathogenic form of E. coli that from time to time kills customers at Jack in the Box or Chipotle. So the non-human side, I think, really needs attention drastically and immediately. And unfortunately, most of the attention has been sucked onto the human side.
But whose job is that? Whose job is it nationally? Whose job is it internationally to do that?
To
regulate this, to make sure that nobody's using it incorrectly.
So
who's there to make sure that if they put the Nexus together and come up with a framework, that it's actually...
So in the US, it's Congress, ultimately. Internationally, good luck. Coming up with broad standards that countries will, by and large, adhere to will be complicated. Getting Congress to do the right thing is difficult. Groups like the National Academy of Sciences, which sponsored the summit that David co-chaired and which came out with the recommendations on human germline work, has a committee currently thinking about regulatory structures for non-human uses of gene editing. But I worry that unless people start paying attention and recognize it's significant, it'll just sort of keep churning over without any progress until something terrible happens. And then Congress will immediately, in an urge to do something, probably do the wrong thing.
I will note for those of you who are Jurassic World fans that on the subject of what other uses gene editing might be put to, George Church at Harvard, whom I'm sure David knows very well, and Hank probably as well, and the whole panel is proposing to bring back an extinct woolly mammoth by deriving genes from frozen corpses, gene editing and injecting them into elephant embryos. So for those of you looking for Jurassic Park, keep your eye on that. Well, I think we are at the end of our session here. Just any closing comments from the panel? I would like to read, perhaps just to tee that question up. David, a quote from you on the germline editing, which is, the overriding question is when, if ever, we will want to use gene editing to change human inheritance. George Church again. On the other side, this technology is poised to transform preventive medicine. Rather than talk about the possibility of banning alteration of the human germline, we should instead be discussing how to stimulate ways to improve its safety and efficacy. Which side of the argument We know we're David since he has the one quote already, but we'll start with you, David. How do you respond to George's?
No, I think you misread my quote. Misinterpreted my quote. I said we should be careful. I don't think George argues that we shouldn't be careful. I think we're both basically in the same place. I think we should move ahead with research. We should move ahead with perfecting the technology. We should deal with the issues of what is appropriate and what is not appropriate as a separate issue, not try to use a technology lock on that. That never works. I do think that the international issue is very important, and there is no way, as Hank said, to have international law on this issue. Rather, we can have international moral force. And that's what we did at Asilomar. We put together something that made such sense that everybody said, yes, we agree. There was no violation of it anywhere in the world. I think we could do the same thing with CRISPR-Cas9. What we have to do is to have very strong, well thought out positions and use opprobrium as our force, not law.
Okay, I think that's an excellent closing statement. And David, thank you very much for your role for decades in setting the tone for debates on this and other controversial subjects. The world owes you a great debt, so thank you very much. So members of the panel, thank you very much. I hope the audience has enjoyed it, and if you're interested in If you want to hear more about Zika and Ebola and that sort of thing, you can stay right where you are, because I think that's the next panel here. So thank you very much, everybody.